if you're online, since we only have like 2 minutes left, I'm just gonna say you're not here. Same as usual, I'm just not gonna look to see who's here. If you're online, just send in the words as you usually would. So Aki is not here, Boggs is not here, Bonner is not here, Clay is not here, Dawson is not here, Ellis is not here, Castro is not here, Green is not here, Harrison is here in red, Henson is here, red, yeah, in red. James is not here, Kesterson is here in red, King is not here, Lewis is not here, Linda Cube is not here, Morrison is not here, Grace is not here, Ward is not here, Wilcox is not here, Wilder is not here, and Parks is not here. And again, I know there's a bunch of you online, but I did get started sort of late, so, and every time we get done, I get an automated list from Google that tells me who's on and who's logged on. It's almost a formality. Anyway, it's right here. So this week, next week, it's fall break. I came back from last week. We're running out of time, guys. Anyway, yeah, sorry. As I'm sitting here, I'm looking at our calendar. So we have, today's obviously the last day of this week. Then we have next week. And after that, we have fall break. Looks like I think we have, one more, I believe one more week of class, and then, finals week. So we're down to our last 2 weeks. So I know I've been saying this a lot, but now's the time to get into your independent work. Because again, it's 100 points out of 1,000. So it's a whole letter grade. Even if you score 100% on everything in the semester, if you didn't do your independent work, you're going to have a B. So, or conversely, if you would have almost failed because you didn't, maybe you had like 500 points, but then you did all your independent work, then that'll put you up to a passing grade because it's worth an entire letter grade. Anyway, Dawson just got here. So let's get started. Any questions about anything? All right, yeah, so get some independent work done. I mean, I'm really going to regret it, especially for those of you, I don't know how to put this. So it used to be, has been, if you turn something in and there's a mistake, I like to coach you on, like, hey, don't do this, don't do that. Make sure you don't use this. This makes it look like you used AI, so on and so forth. I'm trying to coach you. But if you turn this in at the last minute, the last weekend, I've got, which I'm sure most of you will, I don't have time for all that. I'm just going to go through it, grade it. So if you screw up, it's just 0 points for your paper. If you turn in a paper, and you didn't turn it in right, I don't have time to tell you how to fix it. I just got to get through grading all the procrastination papers. So I'm just going to have to tell you, like, that's a 0 for that paper. Let me get on to the next one. So now's the time to work on it. And if you need help, that's what I'm here for. I mean, you have to write it yourself. But I'm still here to help. Like, if you want to talk to me for 5 minutes, like meet me online or something, like, hey, I was thinking about writing about this. What are your suggestions? I'm like, oh yeah, you can focus on this, focus on that. You might want to look this up, avoid that. Let me help you. Anyway, if no one has any questions, let's get back into the material. We are still talking about the evolution of populations. Last thing we talked about was the sources of genetic variation. So we talked about mutations and sexual reproduction. Now we're talking about populations as a unit of evolution. And I've already said this information before, but now we're just really going to reiterate it. And this will be a test question. Don't mess it up. The first word for attendance will be units. Units, like the word I just circled. Anyway, yes. So once again, before I even jump into this main bullet point, I'll reiterate the fact that populations evolve, right? Individuals do not evolve. They might change. Individuals might adapt, they might change, but that is not considered evolution. So evolution only happens on populations. So if you forget anything else I'm about to teach you for this bullet point, remember that. The evolutionary impact of natural selection is only apparent in the changes in a population over time. And once again, what is the population? It's a group of individuals of the same species that live in the same area and interbreed. Wait, have we already talked about this? We have, I'm sorry. Guys, I apologize because I remember this picture now. Yes, we talked about that. Here we go. Analyzing gene pools. This is where we left off. My apologies. But anyway, nonetheless, that's still a good reminder. Populations are the unit of evolution. So populations evolve, individuals don't, so don't miss that. Let's talk about analyzing gene pools. So this is basically what would have been great had we done it before you did the Hardy-Weinberg lab. Because the Hardy-Weinberg, that's what it is. It's analyzing gene pools. So let's imagine a population of wildflowers, as you can see here. Some of them are white, some of them are red. There's no advantage to either one of them. I actually won't even get into that yet. Let's assume that the allele for the red flower is dominant, so big R, and the allele for the white flower is recessive, so little r. The genotype frequencies can be calculated from the allele frequencies. And remember, I wish they would not use the word frequencies almost, because I think most people who aren't into math or science, they think of this more as a percentage or a fraction, or a ratio even. So when I say frequency, that is the proper word, but in your mind, you can think of it as a percentage, right? So, well, I'll come back to that idea. So the genotype frequencies, like I said, they can be calculated from the allele frequencies, and I'll give you an example of that. And of course, there's a caveat for that in parentheses, but you guys don't need to worry about the caveat because it's a 100 level non-biology major class. But this bullet point is only true if the gene pool is stable and not evolving. And we'll touch on that a little bit later. In short, generally speaking, all gene pools are evolving a little bit, but again, we'll get to that later. This is where the Hardy-Weinberg formula comes in. It is used to calculate the frequencies, or for example, the percentages of genotypes in a gene pool from the frequencies of alleles, which is a really fancy way of saying, if we know the percentage or the ratio of big R to little r in this example, then we can calculate the genotypes. And I'll give you an example here. This is what it looks like. So the allele frequencies. So for example, if I told you, and that's Q, so that's a big R, that's little r. So also this is something you need to know. If you're taking notes for the exam, this is important. When we're talking about allele frequencies in the Hardy-Weinberg formula, P represents the dominant allele, and Q represents the recessive allele. That will be a test question. P represents dominant, Q represents recessive. Also, you'll need to be, I'm not going to ask you to use any of these formulas, but when you see this formula, you need to recognize that as the allele frequency formula. And when you see this formula, you should recognize it as the genotype frequency. And it should be easy to remember, because think about it. How many alleles are there? There's only 2 alleles. So if you're going to have a formula for all the alleles, that's going to be the one that only has 2 things in it, right? But when you're talking about genotypes, how many genotypes are there? There's 3. Homozygous dominant, heterozygous, and homozygous recessive, right? You already know that. So now you just apply it to this. So when I ask which one is the genotype frequency, it's like, okay, well, there's 3 genotypes, so it has to be this one, not that one. Anyway, back to this example. If I told you, and someone's going to have to give me an answer here before we move forward. This is pretty simple. If I told you that, I don't know, 80% of this population of flowers has big R, then what percent would have to be little r?
20%.
Good. And again, in my brain, and I hope I'm not making it confusing for you, in my brain, saying percent makes more sense, but technically we're doing frequencies. So instead of writing 80%, 20%, it would be 0.8.
0.2.
And 0.2, good, yeah. So I hope this makes sense. Probably makes a lot of sense to you because you're taking statistics right now, right? So this is like an easier version of statistics. All right, so that's how that works, all right? It's sort of like a percentage. Even here, even though this looks a little bit more confusing because you got things that are squared, and if you're into algebra, you probably would recognize this formula. But anyway, again, let me give you another example. If I said, I don't know, 0.3, or let's call it 30% of the population is homozygous dominant, so big R, and then I said, I don't know, 0.6% of the population was heterozygous, so big R, little r in this example. What percent would the homozygous recessive be?
0.1.
B for 3 and 6 is 4. Excuse me, 9, yeah, well, I had to do the math, yes, correct, right? He was quick with that. Yeah, so 1% would be little r, little r. I hope this makes sense. I probably won't have you, even though it's a formula, I probably won't have you do any math on the exam, but if I did, it would be something really basic like this. Where I said, all right, 0.3 is big R, 0.6 is big R, little r, what percent is little r, little r? I'm not gonna have you be doing the real algebra, which I'm gonna give you an example of that later when you're really using this algebraically, taking square roots and things like that. But anyway, pretty basic. So again, the most important thing, big picture, is you understand when you see this formula, that one up top, you need to recognize that as a formula for allele frequencies. And again, that should be easy to remember because there's only 2 alleles, hence there's only P and Q. And then for genotype frequencies, you need to recognize that as the formula for genotype frequencies. And again, that should be easy to remember because genotypes, there's 3 of them. And then finally, again, you need to recognize P as the dominant allele, Q is the recessive allele, and then down here you can see that P squared would be homozygous dominant, which makes sense, right? Because R, right, R squared, that's what that means. Algebraically, when you multiply and R together, you get R squared. But of course, R represents P in this case, so P squared. And then same here. Then you have the heterozygotes, which would be a P and a Q, right? So dominant and recessive. And then of course, your recessive homozygotes would be recessive, right? So Q squared. Any questions about that? All right, this next bullet point is not from your textbook. This is from the old textbook, so I'm not going to test you on it. I only included it because you might be thinking, well, how is this even useful? Why would anybody use it? So, everything I'm about to teach you from this main bullet point right here, this slide is just an example. So don't worry about this for the exam. But the Hardy-Weinberg formula can be used to calculate the percentage of humans with alleles for certain inherited diseases, for example. We can do a lot of things, but let's focus on this one example. So there's a disease called PKU, which is the inability to break down the amino acid phenylalanine. You definitely don't need to know that, but there you go. That's what the disease is. This is caused by a recessive allele. And we know, medically from people who are tested when they're born, we know that 1 out of 10,000 babies in the United States is born with it. Therefore, we can use algebra and this formula to figure out how many people are carriers. Because remember, this is a recessive allele, right? So this means the Q squared people, those are the homozygous recessive. Those are the people that have it. These people who are homozygous dominant, those people just have 2 dominant alleles. So they don't have it, they're not a carrier. But then if we wanted to figure out how many people in the population were carriers, which are these heterozygotes, we could use the formula. So basically what we're trying to solve for is 2PQ, right? That being said, so let's put it in here. We know that 1 in 10,000 babies in the US has it. So that means this is the number for that. This is the percentage, right? 1 in 10,000 would be that in a fraction, right? So that means that's the frequency, the percentage, if you will, of babies, of people in the population who are homozygous recessive. We know that. So we can use some algebra to then solve for 2PQ. First thing we want to do is get rid of that square. So we take the square root of both sides. The square root of Q squared is obviously Q. The square root of 0.0001 is 0.01. That's how we got there. So again, now we're left with Q. We figured out Q. Now we want to find out P. So we subtract the 1, and that gives us P. And then we know that 1 minus Q. I don't want to spend too much time talking about this because it might confuse you guys. But anyway, we know that 1 minus Q equals P. And we also know that Q equals 0.01. So therefore, 1 minus 0.01 is 0.99. Therefore, we know P equals 0.99. Now we know we have what we need. We have P, we have Q, so we can plug it in. 2 times P times Q, so 2 times 0.99 times 0.01. And if you had a calculator, you can figure that out. And then you can find out that about 2% of the population are carriers. And I know I went through that quickly, and the reason I went through that quickly is because I'm not going to ask you to do this. If I were to ask you to do this, I would have really slowed that down and explained it to you. But I just wanted to show you this is how it's used, right? This is how the Hardy-Weinberg can be used. There's many ways, but that's just one example. So any questions about that? And again, as far as the exam is concerned, that's completely useless. Okay, let's talk about microevolution as a change in the gene pools. This is also relevant to the last Hardy-Weinberg lab you did. So one, you did, yes, you used those formulas, but also we were looking at microevolution, right? You were looking at those fish. The fish species didn't change. You just had like 3 different types of fish. You had orange fish, white fish, and spotted fish. And what you looked at was the change in the gene pool, right? Like were there more orange fish, more white fish, more spotted fish? You just saw how that number changed. Oh, it was microevolution, right? A non-evolving, well, before we get to that, there's some other stuff you need to talk about. I told you earlier that we were going to come back to a concept, so here it is. The non-evolving population is in what we call genetic equilibrium, which is also known as a Hardy-Weinberg equilibrium. I don't even know why we teach this. In my opinion, it's very important academically for people who are going to be studying this later in their academic careers. But for you guys, this will be the last you hear of it. Anyway, I don't think we should teach it, but it's not my opinion, or it doesn't matter. My opinion doesn't matter, so here we go. The textbook talks about it, so do I. So what is a Hardy-Weinberg equilibrium? This is when the gene pool remains constant from generation to generation. So that would be like looking at your fish and seeing that they didn't change, which is surprising. Think about it. The first one you did, there was no reason for there to be a change. There was no sexual selection, right? So it's not like the females like orange fish more than white fish. There was no natural selection, so there was no advantage to being, spotted, for example. There were no mutations, so that wouldn't change the allele frequencies. And there was no migration, right? Nothing was leaving, nothing was coming in. There was no reason for that first experiment that you did for it to change. But it did, right? Because things happen by chance. So that's what, again, what I'm getting at is even though we talk about this Hardy-Weinberg equilibrium, it's more of an academic thing that's important if you're really, really, really studying this, which we're not, because this doesn't actually ever happen. Anyway, so another thing that's relevant, or not so relevant, when a population is at equilibrium, another thing that you know about it is the frequencies of alleles and the genotypes are unchanged, right? So again, when something is at Hardy-Weinberg equilibrium, it's not changing. The gene pool is not changing. Percentages of dominant versus recessive does not change. The percentages of homozygous dominant versus heterozygous versus homozygous recessive, recessive does not change. That's all this is saying. So if I ask you anything, just know what a Hardy-Weinberg equilibrium is. It's just not changing. So because generation to generation change in allele frequency is evolution on the smallest scale, we refer to this as microevolution. So again, that lab that you did, you were looking at evolution. Of course, it was virtual, but it is microevolution. And again, I use the same example I think I've used before. Like if right now on Earth, I don't know, 45% of people have a cleft chin and 55% don't. And then 50 years from now, 30% of people have a cleft chin and 70% don't. That would be microevolution, right? We're still humans. Nothing big has changed. But as far as that, our gene pool is concerned, for that specific allele, that specific gene, that has changed, right? But it's still, again, microevolution. Any questions about that slide? All right. The next word for attendance will be pool. All right. So here we go. That was a good introduction to what I'm about to tell you right here. This will be, as far as the exam is concerned, with the next stuff I'm about to teach you is a little bit more important. Because you're going to need to know the difference between these different types of evolution. You already know natural selection. Now I'm about to teach you some other ones. Genetic drift, which also has 2 subcategories. Gene flow, and then again, natural selection, which we've already talked about, but we'll come back and talk about it again. And finally, mutation. Yep. So again, you're going to need to know the difference between those for the exam. I'll say this happened, that happened. I'll give you some hypothetical example, and you say, oh yeah, that's genetic drift. Or, oh yeah, that was gene flow. Or that must have been natural selection, based on the description I give you in the question. So what mechanisms can change a gene pool? I mean, we already kind of just talked about it when you saw the chapter overview. But natural selection is the most important, but there are also other ones that we talked about, like you just saw on that previous slide, but here they are again. Genetic drift, gene flow, natural selection, and mutation. So let's talk about genetic drift. You definitely need to know this. Genetic drift is changes in the gene pool due to chance. Completely by chance. Just like that first experiment you did in the Hardy-Weinberg lab. When we set up our parameters, if you did it correctly, again, there was no advantage or disadvantage to being a certain color. Being a certain color wasn't going to help you get a mate. There weren't any mutations. There were no fish coming in. There were no fish leaving, right? So there was no reason for it to change. But it does happen by chance, and that is what we call genetic drift. So here's a book example. This is from the old textbook. Here's that same wildflower population. We've got some red ones. We've got some white ones. Again, there's no advantage to being red. There's no advantage to being white. So these are just, it's just a random population. And by chance, when they reproduce, again, because there's no advantage or disadvantage to any of this, it's just random. So it just so happens that these are the flowers that reproduced, right? Not because they're better than the other ones or the other ones are worse than they are. That's just how it happened to happen. So because of that, that's the first generation. And look, you can see the frequency, the allele frequency there. It's 70-30. And then by chance, again, those are the ones that reproduce. Therefore, this is what the next population looks like. And you can see now it's slightly different. We went from 70-30 to 50-50. So this second generation is now even, right? And then they reproduce. And any of these could have survived. Any of these could have reproduced. Again, there's no advantage or disadvantage to being red or white, or being homozygous versus heterozygous, right? And it just so happens that when these reproduced, only those two reproduced. Therefore, the third generation is 100% big R's, right? And again, that was just by chance. That's what we call genetic drift. And you can see it was a big change. Of course, it was hypothetical and made up. Probably wouldn't be that drastic, I would imagine. But yeah, good example. Any questions about that? All right. So for independent work, a question. I'll pose you a question that you can answer for 1 point. Just send me the answer. Give me an example of genetic drift in real life. Because people are studying this, so surely you can find a study where it's like, yeah, we looked, and this population changed in this way. So that's for 1 point. And again, if you want to, you can write a paper about it for more points, where your points are then worth, or based on how long your paper is. But if you want an easy 1 point, just answer that question for me. Anyway, any questions about genetic drift? All right, it's pretty simple, right? It's all by chance. All right, maybe a stupid way to remember this too is, imagine if you're driving around on the snow and it's really slick outside and you start to drift, right? So then you're out of control, right? You have no control of your vehicle because you're drifting. So you might hit something or you might not. It's all by chance at that point, right? It's out of control. You can't control it. So hopefully that'll help you remember drift is chance. Anyway, again, I think that's a pretty simple concept, but let's make it a little bit more complicated so you have a little bit more to worry about. Genetic drift has two subcategories. So I'm going to teach you these two. They're called the bottleneck effect and the founder effect. You're going to need to know the difference between the two, and you're going to need to know that both of them fall under the umbrella of genetic drift. And by the names, I hope it's actually I hope it makes sense. The names are actually very, I wouldn't say common sense, but they make sense. Anyway, let's talk about it. The bottleneck effect. Again, this is an example of genetic drift, but it is by chance, but more specifically, it is the result from drastic reduction in population size. So it's a drastic reduction in population size. Now remember, this is still genetic drift, so this is still by chance. So for example, the human population really dwindled, and I was reminded of this of a video that Kesterson sent me. I mean, I knew this, but it's fresh in my brain now. The human population really dropped after the bubonic plague. And there may have been people who were genetically genetically in danger of it, or maybe immune to it. I don't know. I don't remember for sure. Or even COVID-19, for example. So if you think about it that way, that would not be an example. Because if it's genetics that kept them alive, genetics that dictated who lived and who died, that would not be an example of the bottleneck effect. So when we're talking about the bottleneck effect, yes, you definitely need to know it results from a drastic reduction in population size, but also because it's genetic drift that it has to be completely by chance, right? It can't be because there was an advantage to being red, therefore almost all the white flowers die, right? No, that would be natural selection if there's an advantage to being red, right? We're just saying this has, again, a drastic reduction in population size by chance. And that's why it's called a bottleneck. It's because the population kind of goes through a bottleneck, right? And that's the thing that reduces the population size. So what is a bottleneck? This is something that decreases genetic variability. Some alleles just might be completely lost from the gene pool. And this also results in a loss of individual variation, hence adaptability. So I guess for the exam, the most important thing you need to know is this part right here. You need to know exactly what the bottleneck effect is. And then this part right here is less important for the exam, but more important for like, hey, why does it even matter? What impacts does the bottleneck effect have? So again, for the exam, that's more important. But for understanding why the bottleneck effect is relevant, this is more important. Right? It's generally a bad thing. So here's the visualization from your old textbook. Here we have a bottle full of marbles. I don't know if you can see, but we've got some green ones, some gold ones, and some purple ones, right? And again, there's no advantage. One of them is not bigger than the other. Let's assume they're all the same size and weight and smoothness, right? They just happen to be in there. Maybe the bottle's been shaken up. So then we're going to dump it out, and we're going to reduce the population size, right? So we're just dumping it out. So again, we have this large population, and then for some reason, a lot of the population dies, and that's what we're left with. And by chance, we're left with a lot of purples, one green, and then by chance, all the gold ones are gone. And again, there was no disadvantage to being gold. There was no disadvantage to being green. There was no advantage to being purple. It just so happens by chance, those are the ones that came out. So again, when a population does this, when the population size goes from really big to really small, you're going to lose some. And if it's by chance, that is what we call a bottleneck effect. I'm going to give you a real life example of this. But before I do, does anybody have any questions about the switching? Okay, so cheetahs are a real life example. Cheetahs have experienced at least 2 genetic bottlenecks that we know of. One was in an ice age about 10,000 years ago. So again, you had this large population of cheetahs. And cheetahs back then were just like you would imagine now. They don't like being in the ice, right? Because they live in the savannas of Africa, right? They live in warm spaces. So when things got really cold 10,000 years ago, most of them died. Not because not because some were better than the other. It's just they're not equipped to live in the cold. So some of them made it, some of them didn't. Another one, an even better example, I think, is the second bullet point. In the 1800s, farmers hunted them to near extinction, right? So that was the other bottleneck. I will put an X through this. I'm not gonna ask you about these cheetahs. It's just an example. I will point out, though, I think this second example is a better example of a bottleneck because during this last ice age, 10,000 years ago, we know it experienced the bottleneck, but what we don't know is what if there was some cheetahs, there was something about them that let them, survive better in the cold. In that case, that's a little bit like natural selection, right? Because then there's something about them that allowed them to survive, therefore they survived, and therefore they reproduced. So that I don't like that example, but again, I didn't write these textbooks. This is a great example. For the most part, hunters and humans are killing them. For the most part, they just are what they are, right? A hunter probably could kill any cheetah, and therefore the ones they do kill is completely by chance. Anyway, again, this is the example of how a bottleneck affects, and then once again, let's talk about why that's a bad thing. It's because with so little genetic variability, they've reduced their capacity to adapt to environmental challenges. So again, using COVID as an example, again, simplifying it, assuming it was completely genetic, right? You either have the genes to survive it or you don't, which is not the case, but let's simplify it that way. If something happened to the human population where it just so happened to, I don't know, maybe we go to World War III in a couple of years, right? So, right? I'm just kidding. Anyway, let's hope we go to World War III next year, in the next couple of years, and we start dropping nuclear bombs. And most of the human population is wiped out. It just so happens that some people survive, and it just so happens by chance that the ones that survive are the ones that are really susceptible to COVID, right? That would be a bad thing. So the ones that survived the nuclear blast, that was not natural selection, just by chance, the ones that survived, and just by chance in this weird hypothetical situation that I'm talking about, they happen to be okay when they come, or not okay when it comes to COVID. Therefore, our human population would really be at risk. So anyway, any questions about this example or the hypothetical one that I used?
On my paper, I wrote to you about the CARPS. Can I rewrite it and put it, the example as a bottleneck effect?
That wouldn't be bottleneck.
That
No.
would be?
Because nobody in that paper, it was a good paper. I like that you talked about that because we're going to talk about it later in the semester because it has a better use, and then you can probably rewrite it then. But no one looked at the genetics of those carbs. So they were just killing them, right?
Right.
So there's no bottleneck. We can't tell if it was a bottleneck or not because no one looked at their genes to say, yep, we went from small to large population, and here's how the gene pool changed. You know what I mean?
Yeah.
I mean, had somebody done that study, which they could have like looked at all the carbs beforehand and looked at all the carbs afterwards. That could have been an example, but they didn't do that.
Well, they should have checked to see why there were so many of them.
Oh, well, we'll talk about that later this semester too.
I'm getting ahead of myself.
No, that's good though, because they were invasive, and we'll talk about invasive species and why invasive species are so invasive. Yeah, good question. Anything else about this example of bottleneck? All right, now for the other version of genetic drift, and it is called the founder effect. And hopefully this will be easy to remember too, because it's in the name, the founder effect. This is when a few individuals colonize an isolated habitat. Just like it sounds, the founder effect. So you've got some founders of a few individuals who have founded this new area, or this new isolated habitat. It represents genetic drift in a new colony, and it explains, actually that's the most important stuff for the exam, these first three right here. This is what you need to know for the exam. So it is, in a sense, I'm kind of stretching here, but in a sense, it's a little bit like the bottleneck because your population is going from large to small. But in this case, instead of the population going from large to small from death, it's going from large to small because it's a random group of the larger population leaving and going to a new place, right? Random, has to be random. But anyway, some examples of this, and I'm not going to ask you about. But it does explain relatively high frequencies of some inherited disorders in small human populations. Oh man, I wish Linda Q was here. The Afrikaner population of Dutch settlers in South Africa, I didn't even write down what it was. What is their thing? Huntington's disease, yeah. Huntington's disease and Fanconi anemia. There's a relatively higher frequency or percentage of those diseases in that population than there is for humans overall. The Pingelap people, I think they have a higher percentage of colorblind people than humans in general, because again, if humans left a larger population and went to this tiny island called the Pingelap people went to this tiny island, and it just so happens that the people that went there had a higher percentage of colorblind people. Therefore, the rest of that population as it grew had a higher percentage of colorblind people. The Amish, they have a good example, and I'll let you look this up. Actually, for all these, I'll let you look them up. I was about to tell you, but I won't tell you. You can look them up if you want. So what is I already told you the colorblind people there. So for the Amish, I won't tell you, because there's multiple things that the Amish have. So there's a higher percentage of certain disorders that the Amish have. It's a higher percentage in their population than it is in the other population. And the thing I love about the Amish example is because they were relatively few individuals that colonized an isolated habitat. But what I love about this is they weren't geographically isolated, right? You can see Amish people today, they're around, but they're culturally isolated, right? Amish people generally aren't mixing genes with people who are not Amish, right? Sometimes non-Amish people become Amish. Sometimes Amish people leave and become non-Amish. but generally speaking, they are isolated. But again, I love that example because they're not geographically isolated, but they are culturally isolated. So when it comes to breeding, they are isolated. And then Martha's Vineyard. Again, I won't give you that one. No, I'll give you that one. Martha's Vineyard, the people that founded that just happen to have a higher percentage of deaf people, even not even drastically higher, but slightly higher percentage of deaf people for the people who founded Martha's Vineyard off the coast of, I believe, Massachusetts. And because of that, there's just a higher percentage of deaf people there today. As a matter of fact, that's where ASL started, American Sign Language. So again, those are just examples. And again, I already told you about Pingelap and Martha's Vineyard. But if you want to, for 1 point of independent work, just look it up and tell me what are the conditions that Amish people have at a higher percentage than the rest of the population. And for that matter, again, I only gave you 4 examples. There must be some more. So again, for 1 point of independent work, find me some other real life examples of founder effect. Whether it be humans, which are all the examples I gave you, or otherwise. They don't have to be human. It could be any population. It could be a population of animals or plants. So yes, 1 point, if you just send me an email and say, hey, I looked it up, and here's an example, right? Give me the link. Tell me what it says, like in 1 sentence. But again, if you want more points, if you feel like it, you could try to write a paper for that. Anyway, any questions about the founder effect? All right, so we're done talking about genetic drift. Now we're talking about another one that makes sense by its name, gene flow. It's when genes are flowing, right? Excuse me. The old textbook and every textbook I've ever had calls it gene flow. I'm glad I took a little note here. And remember, I'm teaching out of the old textbook for these 2 chapters. But your textbook calls it migration. And it's the same thing, just call it a different name. Because gene flow is due to migration, right? But anyway, it is another source of evolutionary change. And I guess you could say the definition of it is this right here. The genetic exchange with another population. Gene flow is the genetic exchange of another population. So when genes are flowing in, right, when you have things coming in, or perhaps genes are flowing out, or maybe both. Because of that, this may result in a gain or loss of alleles, depending on the situation. But it does generally reduce the genetic differences between populations. So for example, imagine if you had, what was that? I don't remember which Olympics, but maybe even the last one. They had the basketball players for the United States, I think, were put up on cruise ships. So anyway, imagine like a cruise ship full of basketball players and such, like a bunch of tall people, and they crash and they populate some island and they're never found, but they thrive anyway. So now you've got this population of tall people. And then imagine you have a cruise ship full of like, I don't know, horse jockeys and gymnasts, typically short people. And they crash and land on a different island, and they never get rescued, so they grow up and have their population. So now you have these 2 different islands who've never rescued. Generation after generation, these are getting taller, these people are getting shorter. But then somebody figures out how to make a boat, and they find each other, and they're like, oh my goodness. And then they start going back and forth. Then instead of having a bunch of tall people and a bunch of short people, because they're mixing their genes, eventually things will start to even out a little bit, right? And you won't just have tall people and short people. You start having some middle people. Anyway, if you download this PowerPoint, you can click this link and watch another little short video on evolution, including gene flow, which is the purpose of what we're talking about on this slide. Let's see. The next word for attendance, I won't say it this time. I'm just going to put a square around it. That is the next word for attendance. Just put a little box around the next word for attendance. Are there any slide? That's it for gene flow. It's pretty simple, right? Genes come in, genes go. So are there any questions about gene flow? All right. Again, we're still talking about evolution in general. So that's going to bring us back to natural selection because, again, natural selection is one of the ways that populations evolve. And I know we talked about it before, but we're going to talk about it again and give you some more examples and some new terms. So all these things we're talking about, all these different ways of evolving, it's only natural selection that leads to adaptive evolution. What is adaptive evolution? That is evolution that results in a better fit between organisms and their environment. So in other words, natural selection is the only thing that helps a population, right? Founder effect didn't help. It was all by chance. Who went here? Genetic drift, well, genetic drift doesn't help. It's just random changes. Gene flow could help, could hurt, but it's all not on purpose, so to speak. I don't want to say on purpose, but it's not natural selection. So here's an example the old textbook gave. I don't remember if you knew one did it or not. I'm going to X this before I talk about it, indicating I'm not going to ask you about this example. But a blue-footed booby, and I'll show you a picture of this on the next slide. That's a type of bird. It's an excellent example of adaptations for its home. It has a body and a bill that is streamlined like a torpedo, which minimizes friction as it dives. Actually, I guess I should really quickly explain how they eat. They kind of glide above the water, looking for fish, and when they see it, they dive in as fast and quick, as quick as they can, so they can get to the fish before the fish have a chance to escape, right? So because of that, it will be very helpful to have a body and a bill that's very streamlined, right? So you're very aerodynamic, also very hydrodynamic. On the other hand, they are birds, right? So they can't breathe underwater, and they are going really fast, and they are very streamlined. So it's not good for them to hit the water and go too deep, right? Because then they need to come back up and breathe before they drown. Therefore, they have a large tail that also functions as a brake. So they can get their fish and then stop themselves from going too deep and pop back up and breathe, right? And then also, and maybe I shouldn't have included this here, because everything we have up here, above this, that's a great example of natural selection. Everything down here is a good example of sexual selection, and we're actually going to talk about sexual selection later. But while we're talking about these blue-footed boobies, might as well talk about it. The females of these species prefer males that have bright blue feet over, dull blue feet. If you download this PowerPoint, you can click this link and watch another video about natural selection. But here's a blue-footed booby. So again, it's adapted to its environment, right? It has a form that's perfect for doing what it does, for gliding around in the air and then diving in and getting some food. And really quickly, even though we're not going to really talk about this too much when it comes to sexual selection, again, like I mentioned earlier, the females prefer the bright blue feet. And other than that, there's no advantage, right? As you can imagine, there's probably no advantage to having bright blue feet. It's not going to help them catch food. It's not going to help them avoid being eaten, right? So other than sex, there is no advantage to having bright blue feet. And not that I'm going to ask you this, but generally speaking, a lot of these sexual selection things, even though they don't think about this, a lot of times this is an indicator of health. So when the females are just looking at the bright blue feet, like, yeah, come with me, sir. But really what's happening is it's the healthy ones that have bright blue feet, right? It's the diseased ones and starving ones where their feet aren't so blue. So even though that is sexual selection, if you really stretch it in a way that is kind of like natural selection, right? Because the female is selecting, even though they're not thinking about it, selecting the healthiest mate that they can find. Anyway, any questions about that example, which is just an example? Okay, let's talk a little bit more about evolutionary fitness. I'll give you a term you need to know for the exam. Relative fitness. Relative fitness is the contribution an individual makes to a gene pool relative to the contribution of other individuals. Like, in a sense, you could almost say relative fitness is like how many offspring is this individual producing compared to the other individuals in their population. Sometimes this is slightly inaccurately referred to as survival of the fittest. You probably grew up hearing survival of the fittest. Why I put slightly inaccurately in the parentheses is because it's not all about survival. Like, there could be some organism, I don't know, some lion that was born with some mutation, and because of that mutation, it's impervious to all kinds of diseases, right? So every other lion in that tribe has to deal with diseases and infections, but for whatever reason, this lion doesn't because of its genes. But let's assume that same mutation also makes it sterile. In that case, even though it has the best survival rates, it's still not going to help the gene pool because it's sterile, right? So when it comes to relative fitness, even though that thing is surviving, it is the fittest as far as that's concerned. it's still not contributing to the gene pool because it's just not native, right? So again, this is why survival of the fittest is not exactly accurate, because it's not just about survival. Survival means nothing as far as evolution is concerned if you're not contributing those genes that helped you survive. Anyway, that's what relative fitness is. Any questions about that? All right. I think we can do this pretty quickly. There are 3 general outcomes of natural selection. For the exam, I'm going to give you an example. I'm going to say you had a population, and then after a while, your population changed from this to this. And then, from that description, you need to tell me which of the 3 outcomes they were. So keep that in mind as I explain this to you. And we're going to use mice as an example from the old textbook. If we were to graph the coat color of a population of mice, which we will, I'll eventually show you a graph of that, we would get a bell-shaped curve. Meaning, we would have, and again, I'll show you a real picture of it. Meaning a lot of them are going to be some middle color. Very few of them are going to be completely dark. Very few of them will be completely light. Guess that didn't work. I hate that thing. Anyway, we'll come back to that. Let's talk about the 3 different outcomes. You need to know the difference between directional selection, disruptive selection, and stabilizing selection. And I'm going to talk about it because it's up there on the board, but it'll probably make more sense on the next slide when I show you the graphs. Directional selection is when the population shifts by selecting in favor of one extreme phenotype. So for humans, for example, I think the average human right now is 5'6". So that means in like 100 years, if the average human was 5'9", that would be directional selection because we went from being 5'6", like we went towards the extreme tall. Even the other way around, if humans 100 years from now, the average was 5 foot inside of 5'6", that would be an example of directional selection because we went towards we went to the extreme of short, going that way. Does that make sense to everybody? So when you're going one way or the other. So if you had like skin color, right? If we had in 100 years we had a lot more darker people, that would be directional selection. Or if 100 years we had more lighter people, that would be directional selection. Disruptive selection leads to a balance between 2 or more contrasting phenotypes. So that's when you would have, I guess you could say, fewer in the middle and more of the extreme. So again, like in humans, if the average person is 5'6", and you have very few short people and very few tall people, which is the way it is right now. If in 100 years, you've hardly found anybody who was 5'6", but there was a lot of people who were 6'2", and a lot of people who were 4.8 or 4'8", that would be disruptive selection. We kind of got rid of the middle, but the extremes got bigger. And then the opposite of disruptive selection would be stabilizing selection. So in this case, instead of we have even fewer of the extremes and even more of the in-betweens. So I'll just show you these graphs, and this will make a lot more sense. Here we have this population of mice, right? This is the original population. It has really dark ones, really light ones everywhere in between, but it's the middle ones, that have the most. There's a lot of these, fewer of these, and even fewer of those. Then let's say these mice live on an island, a volcanic island. The volcano hasn't erupted in a while, but boom, it just did. Now everything's covered in black, ash and the lava that's hardened. So now it's an advantage to being dark and disadvantageous to being light. Therefore, after a few generations, we start seeing a shift. There's even more darker ones and even few lighter ones, right? We shifted. And of course, mind you, if it shifted the other way, that would also be directional selection. But in this example, it shifted towards darkness. That's directional selection. Disruptive selection, again, whatever happened, all right, again, volcanic island, right, it explodes, everything's dark, but then let's assume it's also winter, so then snow starts falling. So now it's advantage to being dark and light. Because if you're dark, you can hide on the bare rock. If you're white, you can hide in the snow. If you're gray, you're just screwed, right? So you get fewer and fewer of the middle, but more and more of the extreme. And again, finally, the opposite of that would be stabilizing selection. Now you have even fewer light ones, fewer darker ones, but more and more of the in-betweens. So you need to know, again, in the exam, I'll give you 3 different examples. So for one of the questions, I'll say, hey, the population started here, it ended here. What is that an example of? And then I'll have another question. So the population started here, it ended here. What is that an example of? And then again, the third question, the population started here, it ended here. What is that an example of? Once again, for independent work, for 1 point, if you want, you can look up a real life example of directional selection that has been studied, a real life example of disruptive selection, and a real life example of stabilizing selection. I'll give you a hint. Humans have a big one for stabilizing selection. Actually, as a matter of fact, your textbook, that's probably the example they use, but I'll leave it at that. Any questions about that slide? Perfect. So when we get back on Monday, we'll talk about sexual selection, and let's say sexual is the last word for attendance. That's it. I'm gonna go to my office and be on office hours if you need me. Don't forget to send those words in if you're online.